Good afternoon, everyone. Hello and welcome to this Integrated DNA Technologies webinar, qPCR, 3 Pitfalls and How to Avoid Them. My name is Simon Dunbar and I'll be serving as moderator. Today's presentation will be given by Jin Ngee. Jin Ngee is the IDT Field Application Manager for gene expression, genotyping and synthetic biology products in the Asia-Pacific region. Before taking his current role, he served as IDT's Regional Application Specialist for the Asia-Pacific region. It is here that he proved himself to be an enthusiastic professional with a proven ability to design and deliver training on qPCR assay design and the application of synthetic nucleic acid fragments in molecular biology research. Jin Ngee has a BSc in Molecular Genetics from Curtin University Technology, and prior to joining IDT, Jin Ngee was a laboratory scientist at the Institute of Molecular and Cell Biology in Singapore for over 16 years. The presentation should last around 30 minutes, and following the presentation, Jin Ngee will answer as many questions as possible from the attendees. As attendees, you have been muted, but we encourage you to ask questions or make comments at any time during or after the presentation by entering your questions in the Q&A box. Also, please note that you can expand the presentation slide window for easier viewing. In case you need to leave early or want to revisit this webinar, we are recording the presentation and will make the link to the recording available on our website a few days after the presentation. We will also post the recorded presentation on our YouTube and Vimeo channels. You will receive links to these in a follow-up email. And now let me hand over to Jin Ngee for the presentation.
Thank you, Simon, for that introduction. Thank you all for coming to this webinar. Before I begin, I'd like to shout out to the audience that I assume that all of you out there would have most likely performed PCR or qPCR in some manner. This webinar could serve as a refresher to this technique, and at the same time, for those who are attending and have done this before, maybe you'll just come here to appreciate the knowledge. Thank you all. So to begin, let's go through our agenda. We will cover the 3 pitfalls of doing qPCR in general. So the first one would be when to use qPCR. I've come across many practitioners who are actually using qPCR when there's actually absolutely no need to do qPCR for the research project that they're doing. The second pitfall would be detection chemistries. This may sound really silly, but there are situations whereby the wrong chemistry was used for the project itself. And finally, the lack of controls and validation of the primer or assays that you all receive. And here I'll be covering G blocks and how you could design G block gene fragments or synthetic genes as controls for validation. I would cover as well the wonderful products that IDT provides that enables you to perform your research and your work with qPCR, namely our double quench probes, pre-designed assays as well as I'll cover a bit on custom design and when you should do custom design and our wonderful online tools that are available to assist you. Finally, I would help you select your reporters and quenchers for the project that you require. So the qPCR workflow can be visualized as this. You first collect the sample, isolate RNA. If your sample is RNA, in the case of gene expression, you're isolating mRNA. You perform a reverse transcriptase reaction to make a cDNA, and then you perform your qPCR and you analyze the result. All this would, of course, be simpler if you if the analyte is actually DNA in the first place. As you can see, this is a very involved process, all right? And it helps to decide whether you should embark on using qPCR and integrating it into your project. Now, what is qPCR mainly used for? As mentioned in the previous slide, it is a very involved process. And basically, it is a technique which is very sensitive, and when you need sensitive quantification of nucleic acids. So this primarily falls under gene expression experiments. gene expression experiments. It's critical to determine expression levels to understand gene expression. So qPCR is one of the tools that's required for this. Secondly, sensitivity that's required for these molecular events. You need sensitive quantification as well. This is where qPCR comes in. And for example, RNAi experiments, you're detecting minute changes in expression levels. So you can't use protein detection like Western blots for this purpose because some proteins have very long half-lives. So, and basically RNAi experiments are based on a molecular event. So you should be measuring the mRNA levels. Other things include limitations that you've encountered, especially for the sample involved, like single cell analysis or perhaps FFPE samples. You may want to do an orthogonal assay to verify a DNA array. That's one of the things that you can use qPCR for. In general, whenever quantification of DNA and RNA and sensitivity is needed. So before going on, you're going to ask this question, and you've always have to ask this question when it comes to qPCR. Now, we know it's a very sensitive method, but you have to make the decision to know whether it is required. It is not a replacement for a lot of traditional methods out there. Endpoint PCR and agarose gel analysis is the mainstay of molecular biology and is always relied upon. If you can get away with that, don't do qPCR. Northern blots are another example. You need northern blots, right, for RNA analysis. RNase protection assays, qPCR can't do that, and many more. So explore the other options that are available and to know what you're trying to determine. Remember, for qPCR, one primer pair answers one question. You'll hear me often bring this up. It is very important to know what you're trying to determine. So what is this question you have? Right? This will help you integrate qPCR into your project. For example, a specific exon target. Right? Perhaps there's a break in this exon that reduces expression. You want to know whether, and you should target the exon this exon for quantification. After that is done, you try to decide what sort of chemistry you want to use. Right? Here is a often used comparison diagram between 5' nuclease or probe-based assays versus intercalating dye assays. As you can see, the only additional thing that's in the probe-based assay is that probe. And that probe actually offers a specificity towards that amplicon itself. So it detects the amplicon. Whereas compared to an intercalating dye assay, you only require 2 primers, which lends to its affordability. And it uses an intercalating dye that increases in fluorescence as the amplicon products are increased. So here's a nice comparison. I will not go through it in great detail. Just know that with longer amplicons, especially for intercalating dye assays, you get stronger signals. For this purpose, most design algorithms out there limit the length of your amplicons to roughly about 150 base pairs so that you will not generate excessive signals. But that being said, comparing intercalating dye assays and hydrolysis probe assays, what sets them apart, which is in everybody's mind, is the cost. Hydrolysis probe assays do cost more because of that additional probe that you put into that reaction. However, to decide which chemistry to use, you have to decide based on cost and the data quality that you're getting. I specifically say these two because to determine which chemistry you want to use based on the budget you have is not really effective, because data quality, especially if you're out to publish this findings that you have, all right, the gold standard is always the probe-based assays. So if you have reactions that number less than, let's say, 500, all right, in total, that means all the samples that you're processing and all the targets is less than 500, I would say go for the probe-based assays. You get better data, you get specificity, all right? And there's even better sensitivity as well, right? Intercalating dye assays are inexpensive. I understand that. That is a lot of researchers out there would favor this, but I would highly suggest it to be reserved for high throughput analysis. So anything between 500 to 1,000 samples per target would be preferential to use intercalating dye assays to keep your the cost in control for the project. So the next pitfall I would say that we have to address is not validating your assay or primers. Now, you can't just receive a tube, let's say from IDT, and then the primers and probes are inside. You start your qPCR and voila, you have quantification. All right, it doesn't really happen that way because due to the variability of PCR from reaction kinetics, for example, and another factor in this is the manufactured primers, right? Even for the same sequence, manufactured by two syntheses by the same provider, and even on the same day, there will be some variability. And this variability shows because qPCR is such a sensitive method for quantifying DNA. There's a running joke going around that with all this variability, sometimes you wonder why is it still the cutting edge of nucleic acid quantification. So you have to validate your primers and assays prior to use. So you have to generate a standard curve to use as a reference, and you also determine reproducibility for this, for let's say the primer or the assay that you received. And finally, mainly it's just good laboratory practice to do so. Now, performing primer validation is the first thing you have to do whenever you receive an assay or primer pair. All right, make sure you use broad dilution ranges of greater than 6 logs. All right, some people say you can get away with 5 logs. I personally, myself, I prefer 6 log dilutions. Include replicates to check pipetting accuracy. Always include a no template control, which we call an NTC. And finally, about the template itself, the test template. Now, you should test it on cDNA derived from total RNA. But in the purpose of validating your primers, we recommend using synthetically produced DNA fragments or plasmids for this purpose. This validation would, of course, if you know the kinetics and there's some programs there on your instrument that would actually help you determine this, you will get the E value. Make sure you note this down and put it down onto your tube of primers and probe to signify how the efficiency is going on. Now, if you get into a situation whereby your efficiency drops, right, most highly likely there would be tag polymerase inhibitors in the sample or in the controls that you're having. All right. Now, this is these are curves from a primer and probe validation. So this is, the top one is HPRT, which is labeled with a FAM labeled probe, and the bottom is GUS-B with a hex labeled probe. So the black lines are the singleplex, and take note of this. The red lines are the duplex that is together with when both of these are run together. Now you notice that there's a slight drop in the delta RN, all right, of the duplex reaction as compared to the singleplex reaction. Now, bear this in mind, all right, when you load more primers, as in doing higher plexing. Sorry, that's not supposed to happen. When you're doing higher plexing, your PCR efficiency actually drops slightly. And the more plex you do, it actually goes down even lower. So keep that in mind. So this is nice results you see here. The duplex and the singleplex give a pretty good R-square value, and the standard curve is pretty much consistent. Now, standards and controls for PCR, all right. As mentioned earlier, we favor the use of synthetic controls for validating your primers and probes. All right. In definition, a standard is a sample of known concentration. Right. So these standards should be similar in nature and complexity to the gene of your interest. Bear that in mind, DNA analytes should be used on DNA. DNA standards should be used on DNA analytes. Now here, G blocks are perfect for this purpose. All right. They help you measure your accurate pipetting. You can do serial dilutions of these G blocks, and you can also freeze aliquots to maintain assay consistency. So that means to say you don't need to do the replicate serial dilutions over and over again. Just do it once, freeze them, and use them over and over again to maintain that consistency. Now, but first, what are G block gene fragments? Now, way back in 2012, IDT, I would say is the end of 2012 or the beginning of 2012, IDT introduced the first commercially available synthetic linear double-stranded fragments. Now, when this came out, it opened up a myriad of applications. People were using them for mutagenesis. They're using them as gene synthesis fragments. We make a variety of these fragments, up to 3,000 base pairs, and they're very fast on delivery, right? But the mainstay of it, most of the time I've noticed, is that a lot of customers of IDT actually order G blocks as fragments, gene fragments, as for synthetic controls for qPCR. For more information about G blocks, please visit www.idtdna.com/G blocks. Now, to make a G block gene fragment as a synthetic control cannot be much easier, I would say. So first, determine the sequences of your amplicons, all right, including the flanking primer sequences, all right? All you just need to do is take all of them and just concatenate all these sequences together to form your G block sequence. Now, why do I recommend this method of making a G block gene fragment? Now, G blocks are actually sold by blocks. We don't count by per base. So the first block, which comes at a very low price, starts from 125 bases to 500 bases. So it would be pretty much a waste to actually put in one single amplicon fragment flanked by nonsensical sequences on its sides. So instead of doing that, you can actually stuff as many as 4 amplicon sequences on it. So you have one standard fragment that can be used as a control for 4 different amplicons, all right, or 4 different targets. And if you so desire, you can actually set this up as a multiplex reaction. So this example you see here with all these 4 genes, and here are the results of the same tetraplex from the preceding sequence. All right, so here the practitioner has actually done 3 dilution ranges, all right? And from here, look at the curves. Very interesting, isn't it? All these are different, the different amplicons on one single G block, which means to say that they're all equimolarly represented. But yet, look at the reaction kinetics, all right? There's a certain beauty in this when you look at it, that each PCR reaction is different from each other. No PCR reaction is the same. So you can see from all the different targets, the CQ is slightly different for all of them as well. All right, so it's wonderful to see such results when you do a multiplex PCR. All right. So here we see an example of G blocks gene fragments versus cDNA using our primetime gene expression master mix. So with G blocks, you still get this nice curve in this serial dilutions that you see compared with cDNA. So you can actually substitute them especially when you want to validate your primers and your assay, and then finally use cDNA for your actual experiments itself, right? G block gene fragments have been used as qPCR controls by many practitioners out there. Here are just a few examples that they have successfully published and using G blocks as gene fragments. Yes. Okay. So let's talk about IDT's unique products now that we've covered the main pitfalls, unique products, especially for gene expression and qPCR. First, I'd like to cover double quench probes from IDT. We call them the next generation in quenchers. Just a side note, quenching, many companies out there has come up with wonderful, beautiful names, especially for the quenchers. And all of them do suffer from one thing, all right? No matter how good the chemistry or that molecule is at quenching, all right, they suffer from this problem of having distance away from the reporter. Now, your probe usually for qPCR is dual labeled. So the 5' end contains the reporter, and the 3' end is the quencher. Now, time and time again, we realize in IDT that you have to bring the quencher as close as possible to the reporter in order to have much more effective quenching. IDT has looked into this problem and come up with this idea of, double quench probes. Now, double quench probes and the quenchers itself have very unique names. The first one, which we have actually started developing, is called Zen, and it's placed between the 9th and 10th base on your probe, and then followed by the Iowa Black FQ quencher that's on the 3' end. Now, this quencher at the 3' end is very critical because we do not want to present a 3' hydroxyl group for Taq polymerase to extend. So the quencher actually serves that purpose as well as additional quenching for the reporter. And because we have a nice Eastern philosophy called Zen that's named onto our quencher, for Sci-5, we have one that's named Tau. It's only natural, I would say. All right. So what are the benefits of a Zen and Tau quench probes? From this two amplification curves, you can see the background fluorescence of the Zen quencher, the double quench probe, is actually lower compared with the other traditional dual labeled ones. And if you normalize it, you actually see that there is a slight decrease in the CQ number. So it offers a little bit more sensitivity. So quenching actually, if it's very effective, it actually improves your sensitivity. The same can be said for Sci-5 and Tau with Iowa Black RQ. So you are seeing the same results here. So our double quench probes actually are more sensitive, especially when you use them for assays because of this lowered background they have. Now, you may ask, well, I'm doing singleplex assays. Why would I do that? Well, in the event that you're doing dualplex or maybe multiplex assays, like our customers at TetraCore, right? When they're using dual labeled probes, they suffer bleed over signals, especially from their dual labeled probes, because of this poor quenching that they have. Their assay runs roughly 5 to 6 probes in this multiplex reaction. So with our Zen double quench probes, they've managed to bring that bleed over signal to near 0, which actually improved their sensitivity. So there's one area whereby Zen double quench probes prove to be useful. The second would be when you need longer probes. Now, there are certain areas when you want to analyze. Unfortunately, the complexity isn't that great. So your TM for probe, let's say 25 bases, which I would say is the limit for traditional dual labeled probes, it's not going to cut it because the TM isn't that great. So you're forced to actually increase the length of the probe. So in this case, Zen double quench probes or Tau double quench probes are added advantage because the internal quencher is fixed between the 9th and 10th base. So this actually maintains the quenching distance, and therefore maintains the quenching at an optimal level, regardless of the probe's length. So this is one area whereby it can be of advantage. So why order Zen and Tau probes? Well, firstly, lower background, which improves your signal-to-noise ratio, and that also increases your sensitivity. This is really ideal when you want higher sensitivity for your qPCR analysis. You need longer probes without affecting your quenching ability. And finally, this thing that we have. IDT would love to share this technology with all our practitioners out there. And in the event that the assay that you've developed becomes somewhat important and you want to develop it into a commercial test kit, we do not charge royalty fees for this. Finally, the ongoing debate, pre-designed assays or custom designed assays. Now, IDT have this wonderful thing called prime-time pre-designed assays. We covered nearly everything in the human, mouse, and rat genomes, all right, for nearly every gene that's available there. So prime and probe sequences are provided upon purchase. All right, they're all very well designed, all right, through machine learning methods or models. They're based on up-to-date sequence information, includes all the splice variants that we know of, all right, very low price per reaction, very fast turnaround time, because these assays are actually made when you order them, right? And is available either in tubes or plate format. The other thing is that they're also available not just as probe assays, they're also available as primer-only assays. All right, I stress that these actually help you reduce waste instead of ordering traditionally, let's say, 225 nanomole primers and then ordering a probe separately. Having everything all in one does offer the advantage of getting lower pipetting action, which actually reduces contamination. So this is a wonderful thing we have, especially with our prime-time pre-designed assays. Our assays are indeed better designed. This is a chart, all right, whereby we see the mean CQ values for all the different mRNA targets that we have against competitor A. And with a lower CQ, it means our assay is actually more sensitive because we avoid SNPs, especially for our primers. And again, I emphasize again, we have primer assays, all right, identical performance as our probe-based assays. Just in case you need more reactions, especially for those high throughput analyses. All right. Comparing both of them, all right, the probe assay, which is the one on the left, and the primer-only assay, which is the one on the right, identical performance. We stand by our guarantee, especially with our pre-designed assays, that they would perform with an efficiency between 90 to 110% when tested over a 5-log dynamic range, with an R-square fit value of greater than 0.99. So if you have supporting documents that the assay does not perform to these parameters or these criteria that we have set out, we will replace it with no additional charge. And finally, let's address this. Custom design assays. So why do you need to do custom design assays? Now, as I've said earlier, you can't just receive primers and probes, and then you run a reaction, and you say, I've got qPCR results. So the same thing with this as well. You can't exactly input a DNA sequence as a computer program to find primers and a probe, and then you said, I have an assay. It's not as simple as that. So again, like I said, what's the question you need answered? Remember, one primer pair answers one question. You got to know about your target sequence, which targets you want to do. There's a lot of sequence analysis involved. You got to know about SNPs and also the genomic structure of the target. And on top of that, remember, we have prime-time pre-designed qPCR assays. If your targets are actually human, mouse, and rat, I would say it's much more convenient to order these from us rather than design your own, unless we don't have an exon that we have actually covered, all right? Designing your own requires a lot of applied science and knowledge-driven. So I would say that this would be in the realm of organisms and analytes that are not human, mouse, or rat. Right? You need a lot of sequence knowledge to know how it's going to integrate in your project, and you have to have a lot of design considerations, like, for example, specific exons. All right. Now, custom qPCR assays, we don't have them, but we have tools that will enable you. All right. So IDT has our free PrimerQuest tool. I've used it personally myself. There may be some little tricks that you would require to know, insider tricks, which if you were to check with some people out there, they will know how to deal with it. That helps you design specific primers and probes and even amplicon criteria for your target sequences. So for more information, visit www.idtdna.com/PrimerQuest. There are a lot of extensive videos, which actually brings about all these tips and tricks on how to use it. Please view them on our YouTube channel at IDTDNA Bio. Now, this is just a screenshot of our PrimerQuest tool so that you know how it looks like, right? And finally, you need in silico quality control. Now, what is in silico quality control? You design a pair of primers already, but you need to check how specific they are against the target. I won't explain what is going on in this slide, but basically, you just need to concatenate two primer sequences, not together, but separated by a series of ends. Use that as a BLAST query, all right? Of course, you have to do some manipulation in the program selection. For example, you specifically use BLAST-N for this purpose and change the algorithm parameters. This would actually send you back a list of results that actually determines whether your design primers are targeting your specific sequence. So here's a picture, a screenshot of the in silico quality control. There's a BLAST page that you see over here, all right? And in the following page here, this screen cap actually shows you in yellow, those sections in yellow are the changes you need to do to make this in silico quality control with BLAST run properly. Finally, we try to put everything together, your reporter, your quenchers, everything to assemble this assay of yours. Now, firstly, you need to know what type of instrument that you're using. Now, here's a list of instruments. Most of the popular instruments are listed here. There may be some that's not listed. On this chart here, you would see the solid dot. That's the recommended reporter or fluorophore that's for that channel. And the one with the circle or the O is the one that requires some calibration. And then X, of course, it means that the instrument does not support the dye. Now, for simplicity, for anyone that's out there doing qPCR, I highly recommend you to use the recommended dye for that channel for your instrument. Now, calibrating to a different dye requires instrument technician expertise in most cases. So you have to call the technician in to help you out in that. So I know a lot of us are very busy. We want the machine to run most of the time. We don't want any machine downtime. I highly recommend that you just stick to the instrument, the recommended reporter for that instrument. And then finally, your dye. So now you know which instrument you're using, which dye you're using. So now you select your reporter dyes. So our primetime qPCR assays, which is basically our pre-designed assays, they come in 4 different dyes, basically the FAM, Hex, TET, and Sci5. Now, for custom assays or custom probes, you have a wider selection of dyes that's available. And we also support Arto and Alexa floor dyes. So there you go. And after determining your reported dye that you want to use, you determine what sort of non-fluorescent quencher that you want to use. So here in this table, we recommend Zen for the fluorescein-derived dyes, which are FAM all the way to Yakima yellow. Tau is only reserved for Sci5. Anything in between will be Iowa Black RQ. Right? So in conclusion, avoid applying qPCR into a project that does not need it. So it saves you resources. Like in the first few slides, it's quite an involved process. All right, If you really need that sensitive quantification, then go for it. If there are other alternatives that you need to use, or there are other techniques that are more applicable for it, all right? So it's best to avoid qPCR in this case. Now, probe-based assays are always the gold standard. I mean, if you're a researcher out there, you want to publish your data, all right, it gives you the best quality data and should be attempted whenever possible. Now, in the event that you are in a situation whereby you're analyzing up to 500 or more samples, all right, primer-only assays fit this bill, all right? So remember, cost and the data quality are considering factors, all right, not just cost alone. And finally, always assess the quality of an assay design and any assay that you received from a manufacturer. Be it IDT, I know we have extensive quality controls in place to give you the best possible primers, but still you need to do this validation, all right? So because qPCR can produce variable results, so you need to have all these controls to assess the data that you're going to obtain. All this, you do all this to maximize your reproducibility. And finally, main takeaway message is that IDT, we have unique set of online tools that are, and also cutting edge products that will enable you to obtain better qPCR results. Thank you. That's the end of my presentation.
Thank you, Jin Ngee, for that informative presentation. If you have a question and have not done so already, please type it into the Q&A box and we'll try and get through some of those. Okay, Jin Ngee, we have a question here. I'm going to push it to the slide so the audience can see.
Okay.
So the first question is, can we use G-blocks or cloned fragments as standards for quantification? I believe you sort of touched on this already.
Yes. Okay. G-blocks or cloned fragments, which one is preferred? I would say if you can, cloned fragments are nice because the sequence is really verified. There is a pure clonal population that you're obtaining, especially with a plasmid, all right? And if you have facilities to grow it, you have this unending supply of standards, all right? The main drawback about this is that because you're isolating it from bacteria, it has to be very clean. If you have any bacterial DNA, genomic bacterial DNA contamination, it does interfere with your qPCR results if you're going to use it as a control. G-blocks, on the other hand, offer great convenience. Number one is that even though they're non-clonally amplified, they are synthetically produced. And because they're synthetically produced, they do not involve any living organisms like bacteria to propagate them. So there's no other sources of contamination, let's say from genomic DNA. So both can be used. All right? Clone fragments offer you a lot of economy in the far future. G-blocks gives you a lot of convenience. I say both.
Sure. And another question, Jin Ngee. So I'll just push this to the slide. Are purified primers required for qPCR?
No, purified primers are not required for qPCR or PCR for that matter. Now, this is partly due to the fact that PCR is a very robust reaction. Even for primers with slight mismatches on the priming sites, all right, PCR will still power through. Somehow Taq polymerase just likes to extend things whenever there's even a partial match. So if you have primers that are maybe slightly truncated a bit, not really that great, they would still go through itself. But with IDT, you will get better results because even though the primers are not purified, all right, with more full-length product, your efficiency for your qPCR actually increases. So purified primers are not required. I think that's the end of it.
Great, because around 25, 30 bases. They're probably, with IDT synthesis, we're probably around 75% purity, which is really pure for PCR, right?
Yeah, that's right. Actually, I would say closer to maybe 80%, not 75. Yeah.
Okay, so we have another question. I have several genes of interest to quantify. Should I do a multiplex qPCR or a singleplex qPCR for the project?
That's a very good question. Now, in the previous slide where I showed the singleplex PCR and the duplex PCR. Now you notice the duplex PCR curves, the delta Rn is slightly lower, all right? So the more assays or the more plex you put into a single tube reaction, that delta Rn will suffer. Your efficiency will suffer somewhat as well because the primer concentrations would not be optimal. Now, should you do multiplex for if you're investigating several genes? Now, my recommendation is no, unless you're doing a delta-delta RN delta-delta CQ comparison. Now that you could do a duplex reaction for that because you're comparing the expression levels to a normalized gene, all right? But if you're just trying to quantitate genes through a standard curve, I would highly recommend just doing singleplex reactions. Of course, if you are trying to investigate and target several, let's say, for example, bacterial species, let's say 3 or 4 bacterial species in a certain sample, maybe you could get away with it. But a lot of optimization has to go with it. So for genes, I recommend singleplex unless you're doing a delta-delta CQ comparison. Yeah.
Excellent. We've got 2 more questions if you have time, Jin Ngee.
Okay.
We'll take the next one, which I'll push to the slide. So why is Zen or tau placed between the 9th and 10th base? Why not just next to the reporter?
This is interesting. All right. Back in the days when IDT is researching on the Zen quencher, all of us was sort of how would I say, there's a certain company that's actually marketing their probes and naming it after a video game character, right? That actually munches on tiny little dots being chased by ghosts, all right? And the idea is there that, well, when TAC degrades the probe, it's munching on each individual nucleotide. Now, it doesn't happen that way. Now, when TAC hydrolyzes a probe, what is produced is nucleotides, which is correct, and also polynucleotides. So the probe is fragmented. It is not chewed off bit by bit. It's fragmented. Now, IDT did some tests and to evaluate Zen, and when we did place the Zen quencher next to the reporter, and we extended it all along, and every time we did place a Zen in a position that's nearer to the reporter that is shorter than the 9th and the 10th base, the quenching is still in effect. So you don't get any reporting on the quenching at all. So we have actually determined optimally that Zen or tau should be placed between the 9th and 10th base to maintain this reporting as the probe is being fragmented. Yeah, that's it.
Excellent. And the last question we have here is just one that we probably get quite often is TAMRA considered a quencher? If yes, should we continue using TAMRA?
All right. This is interesting. Now, TAMRA was the very first quencher. That's long ago before the dark quenchers came about. Well, for your information, dark quenchers are quenchers that actually take the fluorescence from the reporter and don't emit it. Now, TAMRA is a FRET pair. Now, this I'm talking chemistry terms now, or rather fluorescence chemistry terms now, as a FRET pair to FAM. So basically what happens is that FRET is a fluorescence resonance energy transfer. So what happens is that FAM would fluoresce, and TAMRA would actually pick up that fluorescence, and in turn it gets excited from that fluorescence. So it takes away that fluorescence from FAM. In turn it gets excited, and emits its own TAMRA fluorescence. So what you're getting is that you're not seeing FAM fluorescence, but if you set your fluorescence meter, you can actually measure the TAMRA fluorescence. Now, I'm not saying that TAMRA is obsolete. Some places they still like to use it. I don't know for whatever reason, but for most purposes, the dark quenchers have actually overtaken TAMRA. They do provide lower background. Remember this bleed over signal that we are talking about. Now TAMRA emits its own signal, right? Its own fluorescence signal. So you can't actually use it for multiplex. So you can continue using it provided you're doing a singleplex reaction. But for all intents and purposes, I would highly suggest everyone to move towards the dark quenchers, especially the Zen and Tau quenchers. Yeah.
Thanks, Jin Ngee. Okay, that's all the time we have for questions. I want to thank you all for attending today's presentation. I'd also like to thank Jin Ngee for his presentation as well as the Q&A session. This is one of a series of webinars we'll be presenting on qPCR and other topics. We will email you about these future webinars as they are scheduled. Also, as a reminder, a recording of this webinar will be posted shortly on our website and at www.youtube.com/IDT-DNA-bio. There you will find several other educational webinars on topics such as genome editing, next generation sequencing, genotyping, and general molecular biology. Thank you again for attending, and we wish you success in your research.